very excited to have our scientific advisors here. We are missing a few from our crew this week, unfortunately, due to some family issues. But we're excited for the crew that we have. So, just by way of introduction, I believe you have this information in your agenda and handouts. Dr. Michael Wangler, you've heard from him already today. Dr. Nancy Braverman from McGill University. Dr. Femke Klouwer from the Academic Medical Center at the University of Amsterdam. Dr. Bose from Montclair State University, also the parent of a GFPD Warrior alum, and Ann Moser, our favorite person in the world. Everyone in this room's favorite person in the world. So I'm going to let Mike start this off. We have questions from our families, and I think we've got some mic runners here. So we should have 2 mics ready. Oh, cool. All right. Let's let them share just a little bit, and then we'll go to Q&A. So thank you.
Okay. Okay, thanks. So I thought I would just, before we go to your questions, which is intended mainly to be questions for families, but there's several of the scientists and trainees here as well. I thought I would first summarize that the group of us actually sort of our meeting started yesterday, and over the past 2 days I think we've heard a lot of exciting things. We started off by hearing more about the Nancy brought everyone who's new to the field up to speed on peroxisomal disorders. We heard about the natural history study. We went through some of the grant mechanisms, the successful mouse grant that Nancy and Joe put in, and we heard about some of the mouse models. And then we had an N-of-1 therapeutics session focused on oligonucleotide therapeutics. We have a sort of guest speaker, Austin Larson, Colorado, and Tim Yu at Boston. And then, this morning we sort of continued with Walter Krischevitz, learned a lot about the NINDS funding mechanisms for bringing, treatments that have started out in the lab, moving those towards the trial, giving us sort of a checklist, a to-do list of what we need to do. I thought what I would do, before going straight to your questions, is I thought I would ask each advisory board member kind of to briefly just say, what something that maybe struck them over the past couple days. And then I think the families, might want to hear our perspective on where we need to go. So, we're advising the GFPD on the direction of the science. So I think just hearing sort of something that struck you, and then where we need to go. Easy.
Well, again, okay. So, of course, I was struck by all of these opportunities that are available, for therapeutic opportunities. Dr. Korsitz gave us a whole bunch of ideas of things that our disease is applicable for and at a stage for and applying for additional grants. I think we have, it's really important for us to be successful in this Center of Peroxisomal Expertise grant that we put in the fall of 2018. Although we didn't make the cut, we did get fairly good scores, and we got some good reviews as to what to focus on. And I think that would be really important because this is not the research side, but this would be on the clinical side, with the natural history studies, with looking at maybe some preliminary clinical trials, and then bringing together patients for proper medical care, which is always talked about. So I thought that was really exciting.
Yeah, I saw it come up with
And, also it was great. It's really great that we were able to participate together. Everybody's here. I heard from some students in the audience that this is a great meeting for people who are working in a research lab and don't really understand what's at the other end with the patients with this disorder, and that it is really invigorating and it provides enthusiasm. It gives them passion to go back to their research in the lab and learn more about these disorders for us to, of course, use as patients. So I think one of the things to think about in future meetings would be to bring in, to set up some travel boards for students to come in who work on peroxisomal disorders and sort of similar types of things to bring more students in research to see the families what they're actually studying, and then additional researchers that are doing peroxisomal work that may not have been to the sleeping or may not know about the sleeping. So I think that would be really great as well.
I guess what struck me is what great advantage has been made, like again in the last few years. And every conference I come here, and I'm of course from Europe, but every time it struck me again that so much progress is made again and again, and yes, always warms my feelings. But again, of course, we also agree at the conference that also a lot of work has to be done in the future. But conference really helps to share ideas, to also be critical, I guess, about each other's idea, which in the end is a good thing because then you come up hopefully with the best strategy. And I really love seeing the families again, but what also struck me in that is that I heard again stories that I think tells us that a lot has to be done, also still in counseling, for instance, of the different entities out there. So that is, I think, what comes to mind now. Thank you. No.
Okay, cool. Can I sit down? Okay, good.
Hi. I think for me, what really strikes me, what I love, and I think this is sort of me having that kind of dual perspective, being both a parent and somebody that researches this disease, is the sort of engagement that our scientists have with our families. We have these discussions where we involve families and really get feedback from them of what do you want to see, and I think that's really what is going to make these clinical trials moving forward meaningful and impactful to our families. And okay, this is real significant change that means something to me and my family. And that's really what the hope is with these clinical trials, is that this therapy is making my family's life better in very specific ways. So that's one thing that that sort of struck me. And then another thing that I really like about this conference is, again, every time we all get together, you always see things moving forward. Probably not as fast as all of us as parents would really like, or as researchers as we really like, but that's sort of how it goes. But it is always moving forward, and that's really nice. And with that moving forward, there's also this sort of acknowledgement of, we are still living in the now. There's things that we need to be doing now to make sure that our families are best taken care of as well. So I think that's really what we need to continue to do, is always look to the future, but also think about what we can do now to make our families' lives better.
So being at this meeting, I realized what an exciting time it is in the field of peroxisomal research. There are many new ways to achieve therapy with the ASOs, gene therapy coming along, and to hear from the young people, to meet the young people, see their posters, realize that we are recruiting young people to work in this field. And that's really heartening to me as a long-time researcher to see this research carried on. And to learn from the Jackson Lab mouse models, there are many different hex gene mouse models now that are available for research. And to meet new families and reacquaint with the old families and see people that I've only seen on Facebook, see them in person here. That's a great advantage for being in person at this meeting. And many thanks to my colleagues, particularly Joe, who's written a grant on the hex gene mouse models, and our lab is now working on the hex-1 mouse model that's receiving gene therapy at the Jackson Labs. So again, it's a very exciting time for research on peroxisomal disorders. Thank you, Jay.
Thank you, Anna. I am just graced to be in the presence of amazing people here, just on the stage. Anna's one of my heroes. My family, just like I love dancing others, that's family. And to have the opportunity to be here with you and to be embraced by you. And it really does touch me and humble, and I feel really humbled. I think if I had a loved one with peroxisomal disorder, I'd be Nancy Braverman's worst nightmare. I cannot believe the grace that you are giving me when we come up and talk about research, and you can tell I've been really trying to pound down timelines and money. It's not the most fun job in the world, but I do so simply with the love in my heart of thinking about someone who has a loved one with a peroxisomal disorder to let them know where we're going, what's needed to be able to push things ahead, and to try to do this as rapidly as possible. So I do believe in the future, and I really respect all the wonderful work that's being done and networking here. Anna is doing a wonderful job with a club called the Peroxi Club, where you can go on Zoom and there's 100 bright and active faces from across the world all excited about peroxisome biology. I'm hoping we can increase the number of medical students and trainees that could be able to attend these meetings to see what a wonderful community there is. I think there's a lot of room for promoting diversity in our trainees and in the medical community in this way. So again, thank you, and God bless, and I hope we have some time to be able to talk one-on-one throughout the rest of the meeting.
Great. And I'll just echo a couple things that I heard. One for me was I think I merged with a newfound excitement for bringing the next generation on. We kind of heard from trainees, and for me it was a realization that if trainees are given the opportunity to kind of find their passion, they are going to enter our field and help change it. And then and actually being at the meeting and interacting with families, a huge thing for trainees. So that was something that I should have known, and then I guess, I would also echo that I feel like two priorities that maybe we need to think about are that bringing in, new researchers and keeping them excited, but then also the therapeutics. Deciding upon which therapeutics we're really going to pursue, with all the and then being clinical trial ready for those therapeutics with the endpoints that we need. So I think what we want to do is open it up for questions. I think we can start with general questions. And it can be directed at the entire board. You could have a question for one of us. Never ask for questions, because it's one. Okay, good. Yeah, I need to chat.
Okay, just to start out. Okay, Joe, so you've been drilling timelines, you've been drilling money. What do you see as the next step, and what do you need from us?
Thank you.
Let me pass that and answer that. Well, this is a good question. One of the first things we have to prioritize is which are the organ systems that we're going to be targeting. If you're going down a gene therapy route, I'll leave this to Nancy in a second about maybe she can speak a little bit about diasmetin, because that's something that's come up several times, and that's her baby. But if we are we prioritizing? We talked about the retina. We've actually got something to work in the retina in the mouse, which is amazing. And we also heard from Catherine, we don't know the exact timeline, but if we do get an investor, probably starting from day one, it's still going to be 4 or 5 years down the line. Okay? Now, one of the limiting steps right now, if you're trying to treat a CNS disorder, is that you need an animal that actually resembles the human condition, meaning a mouse that has some sort of neurological condition. Now, this is where it's just very pragmatic right now that we have an animal that certainly shows retinal degeneration, and that's why we heard in Catherine's amazing work that you could be able to test therapy. They certainly also have liver disease, so you could be able to test therapy for liver disease. Now, if you have an animal that has a normal brain or nearly normal brain functions, that's basically the animal that we have now, well, there's nothing really to fix. How can you even test something? So that's one of the reasons why we're working with Nancy, and especially with the Jackson Labs, on a few ideas. And Miriam Bose is here, and she's like the world's expert on mouse models of peroxisomal disorder. She's basically written a textbook on this. So one of the first things we really want to do if we want to address the CNS is to have a mouse model that recapitulates to some degree what's going on in the central nervous system of the patients. So that would be number one. Then number two, if you want to put the pedal to the metal, and this has been done before, if you have a loved one where there's neurologists would agree that person could potentially benefit from a CNS gene therapy, you have to really get buy-in from the experts. If you have the money, you can actually ram it through. Now, in the old days, I would say the record I saw was 2 years. Now, the problem here is now there's a lot of all these gene therapy trials creating vectors. There's a backlog. And then there's actually another technical issue, especially with PEX1. Now, it's a very technical issue. You're saying, hey, wait a minute, they could be able to put this into the eye of a mouse. It looks like it's delivering the gene. Why don't they just inject a little bit more and get it into the brain? Well, it just turns out something very technical, that PEX1 is a fairly large gene, and you can fit it into the virus. But that virus is kind of a little wimpy about how it actually delivers the gene. You could be able to really infuse like a retina, which is about the size of a dime in a human, far less in a mouse, of course. And you could be able to basically get around the fact that it's a little wimpy and be able to deliver the gene. You cannot do that with a big organ like the brain. So you'd say, all right, well, you need a technological advance to create a virus that is more effective. Now, the nice part is that, having these scientific conferences, and what I've done with Nancy and the rest of the GFPD, is invite outsiders from outside the peroxisome field. Often I feel terrible that we're not getting all the stars of the peroxisome field speaking every year, but occasionally I need to get outsiders. and I think about the technology folks. And I could say, I know folks in industry that are saying that in the next 2 to 3 years, there'll be the next generation virus that could package something the size of Hex1 will be used for tested for another genetic disease. So basically, that new generation virus will be tested in clinical trials first time in human. We don't know if it'll work. It could actually be toxic. It might actually not even work at all. But nevertheless, there's hope because it's a different community of scientists. These are the engineers that think about how do you deliver DNA, all these different delivery systems. And we're tied in, I think, with the best people in the world. But regardless if you're Joe Hacia or you're Francis Collins, and I spend time with Francis and we talk about some of the rare disease groups he works with, you run into the same types of roadblocks. And what I'm trying to do, and others on the panel, everybody's doing, is learning from other communities. It's not just the peroxisomal disorders. You'd ask, why is there a roadblock like treating Huntington's disease or ALS or something like that, and learn from those lessons to propel the research forward. But now I'll give the mic to Nancy to be able to discuss a little bit about diazepam, because I think that's something that people are going to be interested in, correct?
Well, I know it was on the summary sheet of the discussions that we had earlier this afternoon, but we had, this was just in an initial cell screen using fibroblasts from an individual with Hex1 G843D mutation. We identified a class of drugs that could rescue peroxisomal function, and this was further worked on by a student in my lab, Gillian McLean, who unfortunately passed away, and that's why one of the ideas we had was to make an award for her in one of the last rounds. But she really pulled this through and identified from this class a specific flavonoid molecule that had the best recovery of peroxisomal function, and within 2 days we could basically correct the number of peroxisomes there and the import of multiple enzymes into the peroxisome. And the concentration we used was reasonable in micromolar concentration was reasonable to bring over. Clinically, we did not have a mouse model at that time, and so we thought about of going straight to the patient. There is a compound called diosmin, which is an oral prodrug of diazepam, and it had passed through a set of European guidelines for approval in pregnant women for 3 months for varicose veins. But there was no data had been available for children, and we went back to the company to ask them about it. It was Servier. They didn't really want to be involved because they didn't have any data for children, so they didn't want the liability. And at that point, we had the mouse model developed, so we thought that we would try this drug in a mouse model because we know it would recover peroxisomal functions in cells, but the question would be, does it recover vision, does it recover liver disease, which is what we really want to know. And so that's where you have to go back again to a mammalian system. So we did that, but unfortunately the drug diazepam ends up being really, really insoluble. And we just could not put it in a solution so that we could inject it into the mouse before it precipitated out. So we got stuck there. That was before the pandemic. And so now Joe, with his great networking capabilities, has found someone who's a researcher out in California who is working on solubilizing this class of drug called flavonoids, and he offered to help us, and we now have it solubilized. So we're back to square one in that experiment, and hopefully we'll be giving it to the mice and having some information on it. It turns out, though, that the story gets a bit more complicated, as things always do, because this flavonoid gets metabolized by the body very, very quickly. So when it's used in the oral human form diosmin, it's usually given twice a day, and we have restrictions on how much volume we can inject into these mice per size, and these are tiny mice in this model, and so even we don't have the best conditions to, even though we have it solubilized, we still can't give it twice a day, every day, which is what we would like to do to see the best effect. But nevertheless, it's definitely worth trying in the mouse model. We had thought about, still have thought and brought on the table of taking the diosmin oral prodrug and going straight to the patient without skipping the mouse model, and we could have that discussion again, but we would still need approval from the REB for a compound that there's no data for children. We might need to get it approved at the health and FDA level. Give it. We would need endpoints of how to follow it, and so we have to go through we have to put ourselves in the mode of setting up for a clinical trial and deciding how we were going to assess its efficacy. So at this point, my gut feeling would be to still try, since we have it solubilized, would be to try it in the mouse model first.
So I mean, for sort of Corey's question about like what the GFPD could do, I mean, I guess you guys can correct me, but I think it's sort of like for us where we're trying to balance that we want to bring in new scientists. That doesn't directly bring a therapy today, but it's something we need to do to continue new ideas. Then on the other hand, there's things like Catherine's gene therapy where you really do start to see a path now, so we definitely want to support that. And so I wonder if, kind of for them, I guess my thought would be that it's a similar balance where there's, as an organization, GFPD has to support the families, be the primary contact for the families, grow the organization and the group, grow the advocacy of that group, while at the same time maybe supporting some of the gaps. So we heard from Walter Krashevis, some of the pathways that NIH has for bringing those therapies. And I mean, to me, GFPD research support is so helpful for sort of those gaps, right? Where to get you to where you're ready to apply for those NIH opportunities.
I just want to also add that the diosmin brings us back to drug therapy, which remember has a lot of other properties. So when we talk about gene therapy, we're directly into an organ, and when we talk about drug therapy, we can get to many organs. So we also have the GFPD had originally funded a postdoctoral fellow at the NIH to do drug screening, and the drug screening on the PEX1 G843 T cells continued there, and there were additional promising candidates that were picked up and are being evaluated, but we want to restart this now, and Joe has informed me that we probably will have funding to continue that, and that would be really great because our idea would be to put through a drug screen of all of those organizations, even if it's a single one. We would need, we could either create them, we haven't actually talked that far, we could create them in cell lines these days with CRISPR. And if we have available patient cell lines, that would be great also.
More questions from the group?
So hi, this is Patty speaking. I have a question as we go through the different therapies. Do you do certain age groups? So if you're looking at gene therapy, maybe just children and adolescents, and then young adults or older adults that are affected with this. Are there age, or is it?
We haven't broken it down into age at all in any of these discussions. So, you know, if you're thinking of, you know, many clinical trials have inclusionary and exclusionary criteria, and sometimes they're really best done if you pick a cohort of individuals that you think might be the best responders. Or sometimes we've heard by Dr. Horschitz earlier that, you know, picking a group like for retinal gene therapy that might not have the best vision would be better than to try somebody with much better vision who might have more to lose if there was a problem. So, there are reasons to stratify for therapy, but we haven't. There's no reason why anybody couldn't be a candidate. And I think the other idea was to remember is that there's probably not going to be one therapy. It's a really good idea to put our fingers in a lot of bowls, and it may be that we have cocktail therapy like there is for many other disorders because it's multi-system, because there's different systems affected as you get older, because different children have different systems affected. And so I really think that we have to, you know, really be interested in a lot of different things to develop them, because in the end, we're going to need a couple of therapies.
If I could just follow up a little bit what Nancy said when we were talking about eligibility for the retinal therapies, you have to also appreciate that in order for gene therapy to work, you actually have to have cells present. The retina has a whole group of photoreceptor cells. It's kind of like an onion. There's all these different layers. I'm a little bit like Shrek in that regard. I'm an ogre as well, or a joker. In that case, you know, if the cell layers are intact or reasonably intact, you can imagine that you can, you know, improve the activity or the function of those cells. If they're not present, then that type of therapy is not the right option. So that's one of the reasons why in the past, when we've had, you know, meetings where we invited a lot of different speakers, we had investigator Hacia and so he started talking about, what would be a therapy in the future for someone whose retina is very degenerated? Is there any way of getting around that and, you know, having some other therapeutic modality? And that's kind of what I've been pushing pretty hard to. I really want to be inclusive of everyone. It kind of pains me very, very much when I start thinking about people being excluded for any reason. That's really the reason I put that R24 grant in, and that was a burden for me after a while, because it was, when you put in 30 grants in a row and you get rejected 30 times in a row, it is not very, it doesn't really make you very enthused. But I basically strapped myself onto the caboose of the JAX train, and I was, no reviewer will say that the Jackson lab cannot make mouse models. There's, as Starbucks is the coffee, the Jackson Labs is the Mars. So we were able to, you know, get this grant, but the real motivation was to give everybody an opportunity, whether or not you have a PEX1 mutation, 6, 10, 12, 26, etc. Ultimately, it might take a few years, but we can have a model that would be reasonably applicable to your situation. And then at that point, we have given some equity here for people to you know, be able to fundraise and be able to try to find targeted therapies for their vote. That was the whole thing. So I feel very graced and honored that we got that to it. And that only came through teamwork. If Nancy's name was on it, if it wasn't on it, that would never come to pass. I think just signing her name onto it, her name is gold. If it wasn't for a person like Amir, who was coming from outside the field and, you know, taking the time to help mentor me about, you know, mouse biology, which I have known very little in the past, but he's been very kind to now. I think I'm semi-functional in my vocabulary now, and so there's a lot of hope there. And I hope you see that we have a vibrant community that does work together and really does love you, and we're all together with this.
You're talking about, you know, getting some younger people involved, and so like I mentioned earlier, you know, being fortunate to be seen at St. Bose Children's Hospital, which is the teaching hospital at WashU, which is a wonderful facility. What direction could you give me to, share Kenneth's story with the students at WashU? Do I go to the college? Do I start at the hospital? Tell me what to do. Because who would not, I could letter and picture, who would not be able to want to help that face, right?
Absolutely. I mean, that's a great question. I know that I don't know how available it may or may not be, but for instance, at Baylor, there's a medical genetics course for the medical students in which, as the geneticists, there will be opportunity to say invite a family with their loved one, for sort of the medical students to meet and encounter. I mean, I'll say for me, I sort of admitted a patient to Texas Children's at 3 a.m. and it was just the questions that the mom was sort of asking and the sincerity of trying to, find out what the diagnosis was. So there's also just the being willing to interact with the trainees. Well, because I'm sure a lot of people have had that experience in a teaching hospital where it's like the third person before you get to the final, sort of attending. But I think just remembering that those trainees can really be turned on to, the field by their interactions. So I mean, I would seek those types of things out, like at the medical school, for instance. Other ideas?
Yeah, in the hospital, there's often a public relations office, and they have, many hospitals have big development funds too, where they like to promote their physicians and they like to promote their patients, and these are ways that they get returns and improve funding. And so, you could go contact that office, with your favorite physician for Kenna, who's really helped her there, and they might be very willing to do an interview and promote that in their hospital monthly newsletter. We have one in our hospital, and I know that there's been several patients in genetics who have been promoted that way, and that's really helped our department as well. And it helps everybody learn about what the disease is and patients that are being treated and the types of therapy they're getting.
Hey everybody. So just one quick question. I know the treatment guidelines were issued a handful of years ago. I know that was a very collaborative effort. I think Nancy and mostly two of the state authors, but I know everybody was involved in that. I was just curious, and for the newer parents in the room, if you don't know about the treatment guidelines, definitely ask me or any of the other parents. They're hugely important. We give them to a ton of our specialists anytime we check into a hospital or appointment. But just curious from the board's perspective, whether informally or whether it's going to be a formal update or just any trends or things you've seen since those were published that might be in sort of the next version, or just other things that as caregivers we should be aware of from a disease management perspective.
I mean, I would welcome updating the diagnostic section a little bit, because as we talked about with some of the families this morning, at least from my perspective, we're essentially diagnosing a lot of patients who are coming from, for instance, their exome sequencing was run in the NICU for seizures. Not a lot of other thought was put in prior to that sequencing about what the actual diagnosis might be. And then PEC's, variants are identified, and that's sort of the starting point for the family, rather than, sort of maybe more of a diagnostic audit. So I think the diagnostic section is something that I would leave what we had in there, but I would just, temper it by saying now, technology has sort of disrupted the diagnosis to some degree.
I think this is a good time to think about that, because Dr. Rizzo, Mike, and I are up for updating our general pediatrics text on peroxisomal disorder. So it's good to add those things. I think.
Yeah, we also did a bit of work on coagulation in zoonotic spectrum disorders in the meanwhile, and there are also some smaller recommendations that might be very relevant for at least a small group of patients. So I guess something like that could also be updated.
I think like since 2015, or was it 2016 when they were published? In that time, there has been so much stuff that has been published since then about, just the characterization of the disorder altogether. So one of my sort of hopes for the treatment guidelines as a whole is moving from, these guidelines were really kind of expert-led, which is great because we had these experts at the time, but I'd really like those guidelines to be more, more evidence-based, and I think we have now gotten to a point where we have a pretty decent evidence base. We have, we just talked about the discussions about severity. I remember Kevin and Femke's paper on adrenal insufficiency. And like you said, the coagulation stuff. We have evidence now to sort of back up some of the expert recommendations from our whole panel. So I certainly feel like it's a good idea to update it. And then this morning we were talking about the growth curve. There's a lot of things I feel like that we could add. And then, I'm sorry, just all these things are in my head right now. The other thing that I know Nancy and I have talked about in the past is giving a little bit more guidance for our populations that are getting older, and because we really focused on those guidelines on the younger populations. So trying to broaden out what that sort of spectrum looks like, or other things that I would include. I have 10 more things that I want to say, but I'll stop. But yeah.
Yeah, that sounds like we need to write a paper, so. So thank you, that's good. Another good outcome we need.
I just have a real quick question. What was the name of the title of the person at the hospital that you should go and talk to?
I don't think there's a specific title, and it may be different in different hospitals. It was like a public relations department, or maybe Aaron, you could also help that there's a development, a funding. This is the department that's responsible for publishing the hospital newsletter. Or at least it's media relations. There's a development fund office usually. Yeah, there's a development fund office usually in a hospital too that looks into philanthropy for people interested in certain things that are going on in the hospital. And they'll actively look for people who will contribute to a new MRI machine because there's a great dystrophy program or something like that. So there's a lot of names, but it's sort of the same idea: public relationships, funding development, what else did you say? Media, communications.
Okay.
Yeah, sorry.
No, it's good.
I think we can keep going.
Here, one more for you, ready. Is there any research that shows that the efficacy of gene therapy reduces over time, meaning as a patient gets older, it's less effective, or there's a specific age where it has to be administered for it to work?
Right, we're just sort of talking amongst ourselves that, we obviously don't know yet, because beyond Catherine's data in the mice, we don't know yet. The issues that have come up are whether integration of the vector, integration of the gene, and then, whether that, whether production of the RNA from that replaced gene could decrease over time.
I can take this one. It's not the age of
of the person that matters. So as long as there are cells that are the age of the person wouldn't be a barrier. Where there is uncertainty is in the durability of the treatment. That is, for how many years will that vector express without turning itself off, or because you want to indefinitely express. I think there's data now from Luxturna going to 9 years at this point, that there's still durability and efficacy 9 years later in people. There's data in non-human primates longer than that the protein is still there. So typically with an AAV gene therapy, you'll attack cells that don't divide because the gene does not go into the genome. So you go for non-dividing cells that will stay there forever, like the retina, like brain cells, for instance, or some other nerve cells. However, there have now been gene therapies administered for the liver, which those are dividing cells, and there has been good efficacy seen in the clinic. So that's all changing, and there is more and more effort being made in terms of understanding whether you can re-administer. So before we thought that it can only be a one-time treatment, so you can't go for dividing cells because you'll lose your therapy over time. And the reason for that is because the immune response will attack that virus, because now you have become, I guess your immune response has seen it before, so it's like when you catch a cold or that it's a vaccination, mechanism that now that virus will not be able to infect your cells again. But there's a lot of work being done for immunomodulation and seeing if re-administration might be possible, and it actually might be to target cells that would need more than one gene therapy hit. So the state of the field is developing all the time. But age of the person doesn't matter. And there's data 9 years out.
Any other questions?
I literally don't need a mic. I'm so loud. You don't even have to watch my face. Be surprised. I know that you had just mentioned that you guys have been in discussion about the growth curve. I'm not sure if this is something you discussed amongst each other or in the earlier session that I may have missed, but is there a timeline on that? And the reason that I ask is because there have been several parents that I've talked to, like probably 98% of parents have expressed like traumatic experiences at GI and our dietitians and things like that, because they just don't have a clear understanding of what growth looks like in our children and tend to tell us that we're not doing enough. So I'm advocating in a way right now of just saying that would be something that would be so helpful to us as parents, is finalizing something like that and being able to get it out to our providers who are not aware.
Yeah. So, that, as I mentioned before, that data came from our natural history study, even though it's retrospective, and sometimes we don't have all the same data on all the children because the doctors don't order the same exams, etc. Everybody does height, weight, and head circumference, and they do it fairly often. So we have lots of data, and we've also taken the data from our study and we've added, Femke has graciously given the data from the Dutch study, and we have made growth curves. We used a statistician that did growth curves for rhizomelic chondrodysplasia, kind of sister disorder, and so we have the growth curves. We had to, as I mentioned earlier, but because it's so interesting, we had to separate the Dutch from the North Americans. The Dutch were still taller, even though they still had this disease. We had to plot them on their growth curves. But anyway, we have to pull the data together, organize it, and organize a submission. Femke, myself, Hermione, and Christine are planning on meeting during this meeting to sort of organize that. And I don't know, I don't want to be committed to a deadline, but I think is the end of June now, so hopefully we should be able to submit by the end of December.
The other thing, Sheena, that that's going on right now is the diet study, and I'm going to be presenting some preliminary data on that tomorrow. And one of the things that we were looking at was, are these kids meeting their calorie needs based on their size and their age? And overall, what we're finding is that not just with calories, but with nutrients overall, everybody's meeting their needs. There's a couple of exceptions there. So hopefully that information will be useful to give to medical professionals and say, yes, they're it's not that we're not feeding them enough. And hopefully that will be something that will be useful when advocating for, whatever crap you're getting from your medical care providers. Sorry, that was a technical term, an industry term.
Other questions from you all? I mean, in the past when we did this, we also had just medical questions. We went global this time, we went big this time.
I had attended some of the Global Genes virtual programs, and everybody's excited about all the new technology and advancements, hopefully over the next few years. But they did say that repurposing drugs is the fastest method, or drug repurposing. Is that what you were talking about, Nancy? Can you explain that a little bit?
Yeah, so drug repurposing is always a faster route to clinic than a novel, newly synthesized drug or unknown compound, because the drug has repurposed means it has been used before as a drug, and so all of the data is there, in the company's hands, like Merck, on the half-life of the drug and how it's metabolized in the body, and what's the safety level, what's the toxicity level, and what's its application, and what are its side effects. So all of this data that you have to accumulate for a new drug is there and can be reutilized, so you don't have to go through the same steps again. So that's what makes it faster. Repurposing means drugs always have other effects besides what they're marketed for. So it's the ability to find that other effect that happens to rescue peroxisomes. And so when we screen our drugs at the NIH and in all of our drug screens, and people have done drug screens with peroxisome disorders in lots of different countries, a library is always used that contains repurposed drugs because it makes the most sense first. And there are several of those libraries, I don't know how overlapping they are, and then you go, after your repurposed drugs, you don't find anything, you often go to, they're called biological products, and that's where we picked up the flavonoids and other types of drugs that are not having to use as drugs before, but they're known compounds that have been used in different ways. And then finally, you might get to a completely novel synthesis. If you knew like the structure of protein, and you can try to synthetically design a molecule that can dock to parts of that protein and improve its folding, for example, and function. And that's a completely novel molecule that you're going to then have to bring into a biologic system and tell the FDA all of that stuff I just mentioned.
And also, just to get at the heart of the question, so the GFPD funded a postdoctoral fellow that worked at NIH for a few years. I think she left in about August of 2019. And by that time, we had screened, at least in cells from a person who had a G843D over I700 frame shift, all the FDA collection. And there was nothing that showed any appreciable activity. And currently, thiazide, at least in our hands, is the champ. But as Nancy alluded to, there's something called PK, which is pharmacokinetics. And it has to do about how the body absorbs a drug, how fast it metabolizes it, where does it distribute in the body, and how is it eliminated. And so you can have your top hit in a screen, which means something that looks really active in cells, but if your body excretes it really, really fast, then it's not gonna, potentially be prepared. So we will continue to screen, and then as Nancy also alluded, then you get to the next round of, well, let's just screen, millions of potential compounds. But then realistically developing that into a real drug can cost far more than gene therapy. You're talking like $50 to $100 million. You're talking about many, many years. There's paths ahead, but the path is slow and it's expensive. That might be in the long term, maybe the real winner. But in the short term, it's actually, unless you get that real hit off the FDA library, and even when you have the FDA library, what people don't say is that, well, suppose it's a chemotherapeutic agent that's given for an acute disorder for a very short period of time, because your body can't tolerate it that long. Well, if you actually have a disease that's chronic, that is going to be a person living for many years, you can't give somebody chemotherapy every day for the rest of their life. So then that kind of winnows it down further. Now, my own mind from this particular meeting, I missed the talks, but I know the subject matter, these oligonucleotide therapies. It's a little bit like, like with these, vaccines from Pfizer, Moderna, and, DNA or RNA based or so. That, these are shorter pieces of a nucleic acid that can be given to somebody and be able to modulate, the activity of a gene, try to make PEX1 a little bit more active, PEX6 a little bit more active. And it's still, costly, but it is something that, you can imagine could be funded through, a federal grant. And could take a few years down the line, but it's, there's a path forward with that as well. And that's something I think from this meeting that we will pursue. And we're going to try to make it as broadly accessible as possible. There's the N-of-1 trials where there's only one person with that particular misspelling and the gene is applicable to. But we have some ideas about how we can take that general approach and try to help more people, say, with PEX1 or PEX6 variants.
I just wanted to say one thing. The other really exciting thing about this time is that there are lots of different ideas for therapy available. So there's these RNA therapies right now that are in preclinical trials. So instead of giving the wild type or the normal gene back, you can give the RNA for the normal gene back, and it's not a one administration deal because you have to give it more often, but you can then titrate up and down depending on a need, and so that's really attractive. And it happens at the moment that they're starting it. It goes to the liver, and there's certain organs that they know they can get it to. So those are the trials. So we also have these things called nanoparticles. So you could put the drug of your choice in this little nanoparticle, and it could target different organs too. So there's a lot of, this is another thing that makes the field exciting now too. There's a lot of opportunities.
Okay, yeah, so it's
Well, I think
you're,
wait. I'm gonna go to big apology to Abby. Warrior camp just closed, and that's why you see parents, it's not, we want to hear what you have to say, but Warrior camp is closing. But if you could hang just for one quick second. First, I want to thank all of our scientific advisors for just the dedication of having all of your assistants.